Hello, my dear doctors. This is Dr. Azam here, your faculty of anatomy at PrepLadder. Yes, my dear doctors. Now we are actually going to learn about the embryology part. Actually, embryology is seem to be a very difficult topic for many of the students, especially in the first prof. You know very well, out of the anatomy, the entire anatomy is divided to like multiple parts here. Like you have general anatomy, then the gross anatomy part, the majority of that one, upper limb, lower limb, thorax and all. So most of the students will be able to deal with that one. But when it comes to embryology and histology, like I have seen that many students will be actually having a difficulty in this one. But believe me, trust me, my dear friends, out of the entire anatomy, embryology is like one of the most fascinating part, interesting part. Why? Because we have all the things like very conceptually here, and we are going to learn in a very conceptual manner here. Fine. So just be along with me and be patiently listening to me, and I'll make the things very much easier for you with the help of like simple diagrams here, guys. Okay. Yes, good evening, Sankita. Good evening, Pratik. now now first of all, to begin with the embryology, I hope you all know that embryology we can actually divide into two categories here. One will be the general embryology, another one will be the systemic embryology. First you have the general embryology, and then you'll be having the systemic embryology. Now in general embryology, what we are going to learn exactly is about the development in first week, second week and third week. So we'll be learning about the development in the first week, second week, and third week. Up to third week, that will be the general embryology part. Okay. And after that, like we are going to learn about the systemic embryology, in which after third week, the systems will start developing, like your nervous system, as well as the respiratory system, the gut embryology, urogenital system, reproductive system. So all that comes under the systemic embryology. Fine. So whatever we are going to learn in the first week, second week, and third week, that comes under the general embryology part. And right now in this session, in another hardly in a span of like 1.5 hour, I'll make sure that I'll make you perfect in general embryology, my dear friends. I'll make you perfect in the general embryology. Fine. Yes, good evening, Dr. Prashant Kumar. Yes, I'm good. I hope you're also doing good. I'm very excited for this lecture, waiting for many days. Okay, fine, no problem. I'm also very much excited to teach you this one. Fine. So if you have understood the basic planning, now let's get started with the general embryology. So I told you very clearly what we are going to learn in general embryology, what is happening in the first week, then the second week and third week of development. So that comes under your general embryology part, guys. Okay. So with that, first let's get started with the first week of the development, sir. Now you might have seen this picture many a times in your textbooks, everywhere. This diagram will be there about the first week of development, but it looks like very complicated diagram. Don't know what all is happening one by one. So what we'll do is, first of all, we are going to make our own diagram for this one here. Step by step, I'll make you understand how to remember and how to learn about the first week of development. Okay. But whenever we are going to start with any week, whether it is first week, second week, or third week, first thing is you should understand and remember what exactly is happening in that week. Like what are the events taking place in that week? That you should have that clarity first of all. So before I tell you, can anyone tell me like what is going to happen in the first week of development? First week of development में क्या होता है? Yes, my dear doctors, can you please tell me what is going to happen during the first week of development? Implantation will occur. Oh my God, heart is going to form. Cerebral college, lower limb. No problem, no problem. Lower limb will be studying about the lower limb also. Don't worry about that one. Now the important thing that, as I told you, like in the very first week, what is going to happen? You should have a clarity in that one. My dear friends, remember that there'll be two events taking place in the first week of development. If you are having your pen and paper ready with you, please start writing with me. Because in anatomy, the best way to learn anatomy will be by drawing the diagrams and writing the notes along with me. Just simply listening will not help you. Now, in the very first week of development, there'll be two events taking place. Number one, there will be fertilization. So you have to remember very clearly that two events will take place here. Fertilization, and the second one here will be the implantation. Fertilization and implantation will occur in the first week. So as a first prof student, you should have a clarity in your mind what is going to happen in first week, my dear friends. That is fertilization and implantation. These two events are going to take place. Is it okay? Very good, Dr. Anish Ghosh. Implantation will also occur in that one. Is it okay? Now, we come across like thousands and thousands of students. So once I was taking a offline class for NEET PG students somewhere in Lucknow. I hope you know the place Lucknow. So Lucknow is actually known for the students being very much what will say शायराना अंदाज होता है। अगर आपको हिंदी आती है तो वहाँ पर शायरी बड़ी बड़ी कॉमन है। तो वहाँ पर हम अक्सर ज़्यादातर हिंदी में पढ़ाते हैं। और वहाँ पर भी मैंने ऐसे ही पूछ लिया, तो बताइए कि फर्स्ट वीक ऑफ डेवलपमेंट में क्या होता है। तो पहले हफ्ते में क्या होता है ड्यूरिंग प्रेगनेंसी। तो मैंने ऐसे ही पूछ लिया वहाँ पर स्टूडेंट से, और एक स्टूडेंट ने शायराना अंदाज में जवाब दिया, सर पहले हफ्ते में तो मुलाकात होती है। तो प्लीज़ रिमेंबर दिस काइंड ऑफ थिंग्स आर लाइक एक्स्ट्रा करिकुलर थिंग्स। तो मुलाकातें और बात है सर, हम आगे बढ़ चुके हैं, हम मुलाकात से आगे बढ़ चुके हैं। तो याद रखिए पहले हफ्ते में क्या होता है, फर्टिलाइजेशन और इम्प्लांटेशन होता है। क्या होता है, फर्टिलाइजेशन और इम्प्लांटेशन होता है, ठीक है। नाउ द डायग्राम दैट आई हैव शोन यू हियर, it's like a big diagram. So let us do one thing, let us draw this diagram together. Now for example, imagine this one here to be the fallopian tube, and this fallopian tube enters into the uterus here, the fundus, body and cervix of the uterus, and below here there will be vagina. I hope you're understanding my diagram here. I'm just drawing like one half of the diagram here, my dear friends. So imagine fallopian tube, then the uterus having the fundus, body and the cervix, and the vagina below. Fine. Now the first thing is that, can anyone tell me what is the duration of a normal, a regular menstrual cycle, sir? What is the duration of a regular menstrual cycle? Can you please tell me here? A regular menstrual cycle will be for a span of how many days? 28 days. 28 days. And yes, very good, Sangeeta. Exactly 28 days. Very good, Anish. Very nice. Now, remember, exactly on the 14th day of a regular menstrual cycle, exactly on the 14th day, ovulation will occur. The ovum will be expelled out. And this ovum will be actually collected by the fallopian tube. The ovum will be taken up by the fallopian tube here. Now you imagine this one to be your ovum here. And this ovum will be surrounded by a layer, and this layer is referred to as zona pellucida. Ovum will be just surrounded by one layer there. That is zona pellucida, sir. Okay, that is your ovum, and ovum will be surrounded by zona pellucida only. Fine. Now imagine that at the same time, even if spermatozoa is also available, then what is going to happen? Fertilization will occur. Now, what is the result of fertilization? Remember, it results in the formation of a zygote with that male nucleus and the female nucleus present in that one. Now, this zygote, it is also surrounded by the layer that is zona pellucida. Zona pellucida here, sir. So the first point that you have to remember is, what is the result of fertilization? Fertilization results in the formation of zygote. So once the fertilization will occur, fertilization results in the formation of a zygote. Now there are like certain MCQs asked over here in your exams, in your entrance exams also. Fine. So the first thing that you have to know is that, when will fertilization occur? On which day? So before I tell you, can anyone answer me here? Okay, fertilization occurs on which day? Fertilization occurs on which day? Fertilization will occur on which day? Come on, answer me right now. Remember, your answer has to be like, fertilization occurs on day 1 of the pregnancy. The first day of the pregnancy, guys. Fine. Now, it's very simple. It's like common sense. It's not even that difficult, my dear friends. Why I'm telling you it is like common sense here? Why? Because, yeah, this is the problem actually. Like I got certain answers. There are like within 24 to 48 hours, and even on 15th day. The first thing that I was trying to clarify, in most of the classes, the students are like, sir, अच्छा, ovulation occurred on 14th day, तो fertilization 15th day होगा, 16th day होगा. But remember, my dear friends, whatever you're calculating like 15th day, 16th day, 14th day, that is with respect to menstrual cycle. With respect to menstrual cycle. Now, once the fertilization has already occurred there, it makes sense the menstrual cycle will stop. The menstrual cycles will actually stop. It will cease. That is physiological stopping of menstrual cycle. तो ड्यूरिंग प्रेगनेंसी menstrual cycle तो नहीं होता है, तो menstrual cycle वहाँ पर stop हो जाती है, तो stop counting with respect to that one. Make sense? Because जैसे fertilization हो जाएगा, obviously pregnancy is going to begin. तो why are you still counting with respect to menstrual cycle like 15 days, 16 days, 17 days like that? So please remember, fertilization will occur on the first day of the pregnancy. Or vice versa, if someone is asking you which should be considered to be the first day of the pregnancy, of course, when fertilization will occur. Make sense? So remember like that. So which should be considered to be the first day of the pregnancy? The day on which fertilization occurs. So fertilization will occur on first day of the pregnancy. One thing done here. Now, second thing is, after the day, can anyone tell me the location? Where is fertilization taking place, sir? Where is fertilization taking place? Yes, of course, we know that fertilization is taking place in the fallopian tube only, but fallopian tube also has got certain parts. They're like fimbria of the fallopian tube, there is a isthmus, ampulla, there are so many parts of that. So please remember that fertilization occurs in the ampulla of the fallopian tube, sir. Very good, Dr. Anish. Very good, Satyanarayan. Nice. So fertilization occurs in the ampulla that was asked in the NEET PG exam. That is the advantage of learning in our Prof Edge classes over here. So simultaneously, I'll also tell you like what are the questions asked in your entrance exams also. Fine. So two MCQs you have to remember from here, 2 points you have to remember from here. That where is fertilization going to take place? Ampulla of the fallopian tube. Number two, on which day? Day one of the pregnancy. Next one. Now what is going to happen? The zygote will further multiply, and it will multiply and divide. Okay, you know that it will be dividing into 2 cells, then there'll be like 4 cells, then there'll be like 8 cells, 16 cells, like that. So it will be multiplying and almost it will be forming like 16 cells right now. And when there are totally 16 cells, this will be appearing like a mulberry. Now because it is appearing like a mulberry here, therefore this one will be known as the morula. और एग्जाम्स में सवाल ये पूछा गया था कि मोरेला में कितने सेल्स होते हैं? 16 सेल्स होते हैं. Normally when you refer to your embryology books, actually they'll be giving a variable number like 8 to 16, 16 to 32, or 8 to 32, something like that. If you want like one correct answer, remember morula has got like 16 cells. There are totally 16 cells in morula. Now this morula will further multiply, and it will multiply to form almost like 32 cells. I'm not drawing the cells properly here, just try to manage. There are like totally 32 cells here. When there are totally 32 cells, then this is referred to as the late morula or advanced morula. It is known as the late or advanced morula. So the first point that you have to remember is that late morula will have how many cells? 32 cells. Now, two more important points that you have to remember here. Now, what are the two important points to remember? Number one, which stage of the embryo will enter into the uterus from the fallopian tube? From the fallopian tube, which stage of the embryo will enter? Is the zygote entering into the uterus, or is the morula entering into the uterus, or the late morula entering into the uterus? Like which structure, which part of the embryo, which stage of the embryo will enter from the fallopian tube into the uterus? So please remember that it is late morula. Your answer has to be late morula. Actually, we have to draw it again here only, late morula, and then it is actually entering here. But I'm like, why to draw it like double time there? So just try to understand that late morula पहले वहां पर fallopian tube में form होता है, और फिर वो क्या होता है? यूट्रस में enter होता है. तो late morula enters into the uterus. Number one point. Number two, second point that you have to remember here, on which day it is going to enter. Remember, on day 4. On 4th day, it is going to enter into the uterus. So late morula enters into the uterus on day 4. Crystal clear concept. I'm telling you, aram se, nothing to panic. It's like a nice story. Remember it like a story only. Yes. Okay. Ab kya hoga? Now what is going to happen? Remember, these 32 cells are going to rearrange themselves. How they are going to rearrange? These 32 cells are going to rearrange themselves, and how they are going to rearrange here? Yes, remember, they will be forming a layer of cells on the outer side. And this layer of cells on the outer side here is referred to as outer cell mass. It is known as outer cell mass. And whatever remaining cells are there, they'll be present on the inner side. And those cells will be referred to as the inner cell mass. So all the 32 cells are going to rearrange themselves into outer cell mass, and then they will be the inner cell mass. I'll be using the short form as OCM and ICM, sir. Outer cell mass and inner cell mass. Fine. Now remember, even this stage is also surrounded by zona pellucida. Zona pellucida. Fine. So now, my dear doctors, already obviously the question will come in our mind that if you are observing from that time, right from like like a like ovum, the zygote, morula, late morula, I am continuously going on drawing the zona pellucida. Now the question comes in our mind, sir, what is the use of that one? What is the function of zona pellucida? So please remember these functions very, very carefully. Listen to me very carefully here, my dear friends, that zygote and morula, these are actually very much sticky. They are very much sticky. The moment they will come in contact with any surface, they will stick there. अब अगर मान लो कि if the zygote and morula come in contact with the fallopian tube, so what is going to happen? It will get, it will stick there and it will get implanted there itself. That means implantation कहाँ पर हो जाएगा? Obviously implantation will occur in the fallopian tube. Now you only tell me here, if the implantation occurs in the fallopian tube there, is it like normal pregnancy or is it ectopic pregnancy? It is not the regular side, it is not the normal side. So therefore, my dear friends, remember it is actually the ectopic pregnancy and this has to be prevented. That ectopic pregnancy has to be prevented. They should not stick to the fallopian tube. Therefore, what is going to happen? That responsibility will be taken by zona pellucida. Zona pellucida. So zona pellucida will prevent that from getting implanted anywhere else and it will safely take the embryo inside the uterus. It will take that embryo inside the uterus and inside the uterus only implantation will occur, not anywhere else. So what is the function of zona pellucida? Function number one. Please remember that it prevents ectopic pregnancy. Zona pellucida is going to prevent ectopic pregnancy. Number one function. Then number two function is that it is also going to prevent polyspermy. Now, what is the meaning of polyspermy, my dear friends? Remember, ovum has to be fertilized only and only by one spermatozoa, not more than that one. So if one spermatozoa is fertilized, then the further other spermatozoa will not be allowed to fertilize that one. And that is also taken care by zona pellucida. So please remember that zona pellucida is a very important structure. It has got like two important functions. Number one, preventing the ectopic pregnancy, as well as it is also going to prevent the polyspermy. Fine. Now, why I'm telling you about all these things right now? Right now, why I'm telling you, sir? Listen conceptually here. मैंने आपको क्या कहा था? What did I tell you? Like what is going to happen in the first week of the pregnancy? What should happen in the first week of the pregnancy? There are two things that should happen: fertilization and implantation. And already we can clearly see here, fertilization is already done. Fertilization तो already हो गया है. Now, what is the second step to occur? The second step or the second thing that should happen here is implantation should occur right now. Oh, तो अगर implantation होना है, if implantation has to occur here, now you yourself tell me with common sense, if implantation has to occur, then what should happen then? What should happen? Who is preventing the implantation? Who is preventing the sticking of that embryo? Of course, that is nothing but zona pellucida. So therefore, now the second step, if implantation has to occur, that means the zona pellucida has to be gone. So therefore, the zona pellucida will degenerate right now. The zona pellucida will degenerate right now. It has to degenerate right now. It makes sense now. That is why I'm telling you embryology is like very conceptual, nothing to be like mugging up, sir. Now the zona pellucida will degenerate. And then what will happen? The ovum will come out of that one. Sorry, the embryo will actually come out of that one. So if the embryo is coming out of that one, it's like hatching. तो इसीलिए आपके बुक्स में लिखा जाता है कि इसको कहते हैं zonal hatching कहते हैं. What is that known as? Zonal hatching. Fine. So zona pellucida will now degenerate. Zonal hatching will occur. Now what is going to happen is outer cell mass, no problem at all. It will be there itself. आउटर सेल मास तो वहीं पर होता है, आउटर सेल मास को कोई दिक्कत नहीं है. बट व्हाट विल हैपन विद द इनर सेल मास now what will happen, the moment zona pellucida is gone from here, the fluid will get the chance to enter inside. Sir, which fluid will enter? Obviously we are in the uterus right now, so therefore uterine fluid will enter inside. If the uterine fluid will enter inside, then what is going to happen? It is going to push the inner cell mass to a side. So therefore the inner cell mass will be pushed aside. Okay. And once the inner cell mass is moving aside, now we can very clearly see this one as a fluid filled cavity. Now in our entire MBBS, you know very well, wherever we come across like fluid filled cavity, that is nothing but known as cyst. Fluid filled जो cavity होती है ना, cavity filled with the fluid, that is known as what's a cyst. So therefore now this one will be known as the blastocyst. Is it making some sense now? इसीलिए उसको क्या कहा जाता है, ब्लास्टोसिस्ट. एंड रिमेंबर, सिस्ट इज लाइक फ्लूइड फिल्ड कैविटी, एंड ब्लास्ट, wherever you come across the word as blast, okay, it means the cell still have the ability to multiply. I think you also have learned about erythroblast, है ना. तो एरिथ्रोब्लास्ट का मतलब क्या होता है, दो cells are still having the ability to multiply. After that multiplying, they will be forming the mature cells which are known as erythrocytes. So similarly, wherever you come across this word as blast means the cells are still multiplying. So that is why blastocyst. Learn with the meaning, my dear friends, that everything will make sense. Okay. So therefore, what is going to happen? I'll just repeat it quickly here. First of all, the zona pellucida will undergo, yes, degeneration, that is known as zonal hatching, and then the fluid will enter inside. Once the fluid will enter inside, it is going to push the inner cell mass to a side, and that becomes like a fluid filled cavity, and that is why it is known as blastocyst. And now you can very clearly see there, blastocyst does not have zona pellucida, so therefore that is going to get implanted. अब क्या होता है सर, implantation होता है. Are you understanding me? So therefore, what is going to happen? Now implantation will occur. That's all, my dear friends. Fertilization done, implantation done, and we are done with the first week of development. इतना आसान है. तो एनी टाइम इफ यू हैव टू रिवाइज द फर्स्ट वीक ऑफ डेवलपमेंट, प्लीज लुक एट दिस डायग्राम हियर. ये डायग्राम अगर आप देखोगे, दैट्स ऑल, यू विल बी एबल टू रिवाइज एवरीथिंग सर. द फर्स्ट देयर विल बी zygote, then there'll be morilla, then late morilla, then it is going to become blastocyst that will get implanted. Crystal clear concept. That's all. And yes, of course, in your prof exams, you can even draw this diagram also. That is the reason why we are taking these classes here. No? You can even now draw this diagram also. And mainly when the examiners are going to check your papers, my dear friends, mainly remember, if you are representing your diagram very clearly, no one will worry about what you're writing there. Why? Because if you are representing the diagram very correctly over there, examiner will understand that ये बंदे को English तो आती है, तो वही तो लिखा होगा. एग्जैक्टली, समझ में आ रहा है. तो एनाटॉमी पढ़ने का बेस्ट तरीका और आसान तरीका, वेरी इजी टू लर्न एनाटॉमी सर, प्रैक्टिस डायग्राम्स अगर इजी हो जाएंगे तो यू आर लाइक 80% ऑफ योर वर्क विल बी डन सर. यू जस्ट हैव टू लुक एट अ डायग्राम एंड कीप ऑन राइटिंग व्हाट इज हैपनिंग वन बाय वन. अंडरस्टैंडिंग? तो आई होप द फर्स्ट वीक इज वेरी क्लियर, माय डियर फ्रेंड्स. जस्ट गिव मी अ थम्स अप इफ दिस इज लाइक क्लियर टू ऑल ऑफ यू. If the first week of development is very clear to all of you, just give me a thumbs up there. अब हम बढ़ेंगे आगे. Now what we'll do is, yes, because we have completed with the diagram here, now once let us even write down what is happening on day 1. On day 1 there will be fertilization taking place here. On day 1 of the pregnancy, fertilization will occur, and fertilization will result in the formation of zygote. And remember, zygote will further multiply, and it is going to multiply to form morula. And morilla will have totally how many cells? 16 cells. And that morilla will now further multiply to form the late or advanced morilla. And late or advanced morilla will be having 32 cells. Okay. And now, my dear friends, the late morilla will now enter into the uterus. On which day? Day 4. Another very important landmark. On day 4, it enters the uterus. Now, once it is going to enter into the uterus, my dear friends, enters uterus. Once it is going to enter the uterus, finally it is going to become blastocyst. It is going to become blastocyst. Okay. Fine. And now, my dear doctors, remember this blastocyst will be very much important here. Like there are so many questions about that one. Let me tell you all the questions one by one. Sir. Okay. So now about the blastocyst, what all questions you have to remember here. Number 1, blastocyst is the stage of implantation. Yes or no? You can very clearly see in our diagram, out of all the stages, zygote, morilla, late morilla and all, which one will undergo implantation? Blastocyst. Blastocyst will undergo implantation. Next, question number 2 here that you have to remember is, on which day the implantation will occur? Implantation begins on which day? So please remember, implantation begins on day 6th. 6th day. मैंने आपको क्या कहा था already? I told you very clearly. The late morilla is going to enter into the uterus on day 4th, and finally what is going to happen on day 6th? It is undergoing implantation. On day 6th, it is undergoing implantation. Now this implantation process will complete by which day? So please remember, it is going to complete by 10th day. Implantation begins on 6th day, and then finally implantation completes by which day, sir? By 10th day. 10th day. Fine. And then where exactly implantation will occur? You also have to remember the site of implantation. Now, can anyone tell me what is the site of implantation, my dear friends? Where exactly the implantation will occur? Remember, implantation will occur exactly at the junction of fundus and the body of the uterus. Okay, na? So what is the exact site of the implantation? At the junction of fundus and body of uterus, on which wall? On the posterior wall. That is extremely important. Extremely important, my dear friends. So that is the exact site of a normal pregnancy. So remember that blastocyst will get implanted at the junction of fundus and body of the uterus. On which wall of the uterus? On the posterior wall of the uterus. On posterior wall of the uterus. Perfectly done, sir. So if you remember these 3 points over here, then you are done with the blastocyst here, my dear friends. Each and every point regarding blastocyst is covered here. So what is the stage of implantation? Blastocyst. Implantation begins on which day? 6th day. Completes by which day? By 10th day. And please remember the exact site of implantation. It will be occurring at the junction of fundus and the body of the uterus. On which wall? Posterior wall. Okay. Yeah, Satyanarayan, it is absolutely correct. It will be getting implanted in the endometrium only. In the wall of the uterus, you are having 3 walls: endometrium, myometrium, and then epimetrium. So you will be actually having the 3 walls. Endometrium में तो होता है, लेकिन exactly site याद रखना होगा कहाँ होता है. So at the junction of fundus and the body of the uterus, on which wall? Posterior walls. Fine. संगीता is writing there as sir implantation नहीं समझा थोड़ा. Implantation यहाँ ज़्यादा कुछ नहीं है. See, I told you, for implantation to occur, what should happen? The embryo should actually stick to the wall of the uterus. But who is going to prevent that one? Who is preventing this sticking to the wall? That is zona pellucida. The first zona pellucida has to be gone. That is nothing but zonal hatching. Hatching किसे कहते हैं? Like, hatching of the chick coming out of the egg, तो hatching जो होता है ना, तो ऐसे मान लो कि आप zona pellucida is completely gone, degenerated, and the embryo is coming out of that one. Zonal hatching. And then because of the hatching, what is going to happen? Now the uterine fluid will enter. And once the fluid will enter, it will push the inner cell mass, this inner cell mass, to a side. And therefore it is known as what, guys? Blastocyst. It is like a fluid filled cavity. Fine. So I think it is very clear now. Hello my dear friends, is it clear now? चलिए बढ़िया सर. इस तरीके से फर्स्ट वीक हमने क्रिस्टल क्लियरली कंप्लीट कर लिया है. वी हैव कंप्लीटेड द एंटायर थिंग क्रिस्टल क्लियरली. Done, done, done. Okay. And now let us move on to the second week right now, sir. Second week of development. And my dear doctors, I'm telling you very, very clearly here. Listen to me very clearly here. Embryology is not like other subjects. For example, if you're learning upper limb, lower limb, and all, when you're learning like upper limb, lower limb, and all, so in those topics, what is going to happen? Every topic is like separate topic. Like brachial plexus, one separate topic. Study, khatam, finish. Cubital fossa, separate topic. But embryology is all about continuation. It's all about continuation. ऐसा नहीं है कि फर्स्ट वीक हो गया चलो भूल जाओ. इट इज़ लाइक कंटिन्यूएशन. तो यू शुड नेवर एवर लीव और लूज दैट कंटिन्यूएशन. अगर आपको कहीं पर भी बीच में नहीं समझ में आएगा ना, ऑटोमेटिकली फिर आगे नहीं समझ में आएगा. If anywhere in the middle you miss that continuation, then automatically you will not be able to understand what are the further changes taking place. So please remember it like a nice animation, nice story. Okay. So now, my dear friends, after completing first week, right now the embryo is in which form? The embryo is in the form of blastocyst. Don't forget that. That is what I'm trying to tell you. अब ये blastocyst का आगे क्या होगा? अब हम देखते हैं second week में. Now let's enter into the second week of development. Now in second week of development, please remember there are like so many changes taking place. So many changes will be taking place in the second week. But I'm here to make it easy for you people. Okay. So as a teacher, we actually come between you and your textbook to make the things easy in the textbook, है ना? तो अब सेकेंड वीक में एक्चुअली होता क्या है? देयर लाइक सो मेनी चेंजेस टेकिंग प्लेस. बट जैसे मैंने आपको कहा है, वेन एवर यू आर गोइंग टू बिगिन विद एनी वीक, ऑलवेज मेक अ हैबिट ऑफ writing down like what is happening in that week. You should know what is happening in this week. So remember, in the second week, there will be formation of bilaminar germ disc. The formation of bilaminar germ disc in the second week. Clear? So in the second week, there'll be formation of bilaminar germ disc. Okay, not the trilaminar. I hope you all have heard about the three germ layers, है ना? The three germ layers, ectoderm, endoderm and the mesoderm, will be formed in third week. It's very much symmetrical here, है ना? तो third week में तीनों germ layer form होते हैं, but in second week there will be bilaminar germ disk. Now, as I have shown you the picture here, there are like so many changes taking place here. अब यहाँ से confusion start होता है, तो I request all of you people to be like, please 200% be alert here. But even though like many changes are taking place here, don't worry my dear friends, we are going to make it easy with the help of 4 diagram. In second week, I'll give you like 4 diagrams here. In 4 diagram, everything will become damn easy for you people. It will become like damn easy for you people, sir. ठीक है ना? 4 diagram में हम सब कुछ खत्म कर देंगे. So मेरी बात आपको? तो please draw all that 4 diagrams one by one along with me. Are you ready? Come on sir, I told you that all the planning here. It's all about second week my dear friend. Second week आपको अच्छे से पढ़ना होगा. It is going to take some time. 15 से 20 मिनट का टाइम लगेगा. I'm telling you all the disclaimers before itself. But why? Because when it comes to third week, again वो एक 5 या 10 मिनट में खत्म हो जाएगा. It will be done in 5 to 10 minutes. Okay, तो it's all about learning second week. Okay, चलिए स्टार्ट करते हैं. तो मैंने बहुत बोल दिया आपको. अब कॉन्सेंट्रेट करना आपका रिस्पॉन्सिबिलिटी है. प्लीज कॉन्सेंट्रेट हियर राइट नाउ. अब सेकेंड वीक में माय डियर फ्रेंड्स, पहले तो मैंने आपको क्या कहा था? आई टोल्ड यू वेरी क्लियरली दैट यस वी हैव टू अगेन स्टार्ट फ्रॉम वेयर ओनली द कंटिन्यूएशन. तो इन द एंड ऑफ द फर्स्ट वीक यू हैव सीन देयर वाज ब्लास्टोसिस्ट. Now, what is the condition of the blastocyst? How is the blastocyst looking like? Blastocyst will consist of outer cell mass, and then there will be inner cell mass, and then you'll be having the fluid filled cavity. Outer cell mass, inner cell mass, and fluid filled cavity. Now, this blastocyst will undergo what? Implantation. It will undergo implantation. I told you this is happening in first week only. I'm just drawing this diagram again so that you remember the continuation. After the class when you're going to revise, you remember the continuation. Ab blastocyst ka kya hoga? What will happen? Now the moment that blastocyst will get implanted, implantation jaise hota hai na, outer cell mass is going to become trophoblast. Let me draw it again. This outer cell mass, it is now known as trophoblast. The moment you are going to write down the names there, my dear friends, I request you to repeat the names in your mind and learn it right now. Learn it right now. What is that one? Trophoblast. So the outer cell mass is going to become what? Trophoblast. Actually, that outer cell mass is like a layer of cells, and we have to actually draw it like a layer of cells only here. I'm just drawing it like one black colored line there. Try to understand that. Now, if you keep on drawing all the cells there, it will take a lot of time. So I'm just trying to give you simple diagram. Let us understand the concept a lot here. So that outer layer of the cells is going to become what's a trophoblast. Then what about the inner cell mass? Now inner cell mass, for example, I'm drawing in blue color here, the inner cell mass here. See how much time it will be taking to draw all the cells, So मान लो कि this one is your inner cell mass here, sir. Fine. This blue colored one. Now this inner cell mass, what is going to happen? Only one layer of this, which is towards the cavity. Observe this very carefully, my dear friend. Only one layer of the inner cell mass, which is towards the cavity here, sir. Which is towards this cavity. Only that one layer will be modified. Those cells will be modified into cube-shaped cells. This one layer only is modified into cube shaped. What I'm trying to tell you is that not all the cells of inner cell mass. No. Only one layer is modified into cuboidal cells, and this layer is now known as the hypoblast. This one will be referred to as hypoblast. Hypoblast. Okay. And this hypoblast is the one, within few minutes I'll prove you that hypoblast is the one which is actually going to become your endoderm, which is going to form which germ layer? Endoderm. So if hypoblast is going to become endoderm, can I call this one as the primitive endoderm here? Yes, of course, I can call it as primitive endoderm. Why? Because hypoblast is the one which is going to become your definitive endoderm. It is going to become the actual endoderm. So therefore, now I can call it as primitive endoderm. Endo, please remember that, not ectoderm, it's endoderm here. That's all. Now, automatically the next question in our mind will be, sir, then what is going to happen with the remaining cells right now? द रिमेनिंग इनर सेल मास का क्या होगा, और वो हम देखेंगे अगले डायग्राम में, इन द नेक्स्ट डायग्राम, डायग्राम नंबर 2. आई होप यू आर ड्राइंग विद मी. नाउ लेट अस गो अहेड विद द फर्दर डेवलपमेंट. अब आगे क्या होता है, डायग्राम नंबर 2. Now, my dear friends, again the same thing. Can you please tell me what is this layer here, outer black colored line? This outer black colored line here, this one will be the trophoblast. Trophoblast. Fine. I'm going to use the same colors till the end of this topic here, sir. Okay, now the outer black colored line is trophoblast. Then I told you that there will be only one layer modified into cube-shaped cells. There's only one layer which is modified into cube-shaped cells. This layer will be the hypoblast. And this hypoblast, please remember that it is also referred to as primitive endoderm. Hypoblast is also known as primitive endoderm. Write the name once more. It is worth writing. You'll be learning that one. ठीक है ना. अब हमारा सवाल यहाँ पर ये है. Now our question here is that कि out of this inner cell mass, only one layer is modified into the cells. Then sir, what about this remaining cells? What about that remaining blue colored cells? इनका क्या होगा? Now those blue colored cells are going to rearrange themselves. And how they are going to rearrange, sir? They are going to rearrange in the form of tall and columnar cells this time. They are not cuboidal, like tall and columnar cells. And this layer, it is present above. तो अगर नीचे होगा तो उसको हम कहते हैं हाइपो, और अगर ऊपर होगा तो उसको हम कहते हैं एपी. एंड दिस वन इज गोइंग टू बिकम एपीब्लास्ट. ओ माय गॉड, इट्स सो सिंपल. Epiblast. Okay. Tall and columnar cells, that is going to become epiblast. And epiblast is the one which is going to become your ectoderm. And because this is going to form your ectoderm, so therefore I can call it as primitive ectoderm. Primitive ectoderm. So hypoblast is going to become endoderm and the epiblast is going to become your ectoderm. So therefore the epiblast is actually referred to as your primitive ectoderm, sir. Whereas hypoblast is referred to as primitive endoderm. I hope it is very clear now. Hello, my dear friends. I hope it is very clear now. I hope I'm not confusing you. I told you very clearly, have some patience there. थोड़ा सा आपको ना सब्र रखना होगा. Second week में, second week will take a little bit of time to complete. Now let me do one thing. Let me check myself. You people are not telling whether it is clear or not. I'll ask you one question. Please answer me. Can you please tell me here, trophoblast will be derived from where, sir? Come on. Trophoblast, it will be derived from where? Trophoblast is derived from where? Let me see whether you are remembering the continuation or not. Come on, come on, come on, come on. I'm waiting for you. Remember the trophoblast, I told you it is nothing but formed from where? Outer cell mass. So trophoblast will be derived from outer cell mass. Fine. Whereas this epiblast and hypoblast, these two will be derived from where? You can clearly see there. One layer is going to become your hypoblast and the remaining cells are going to form your epiblast. So which cells I'm speaking about? I'm speaking about the inner cell mass. Oh, so you should have this clarity in your mind there. The outer cell mass is actually going to become your trophoblast, whereas the inner cell mass, it is going to form your epiblast and hypoblast. Epiblast and hypoblast, my dear friends. Okay. So outer cell mass is going to become the trophoblast, whereas inner cell mass is the one which is going to form your epiblast and the hypoblast. My dear friends, can you please tell me here, we are in which week right now? We are right now in which week of development? Come on. We are in which week of development right now, sir? We are in the second week of development. And I told you in the very beginning itself, in the second week of development, okay, during second week of development, what is going to happen? There is a formation of bilaminar germ disc. Bilaminar germ disc will be formed here. And you can very clearly see in my diagram here right now. Yes, there is a formation of two lamina. There is epiblast and hypoblast. दोनों भी form होता है sir. Bilaminar germ disc will be forming here, guys. Clear? now now after this what is going to happen? Further development, sir. Shall we go ahead now? अब ये अगर क्लियर हो गया है आपको तो अब चलिए आगे बढ़ते हैं. Now the epiblast will multiply. I'm drawing with the same blue color there. Epiblast cells will proliferate. They will multiply to form a layer of cells here. And this layer of cells which will be forming here will be known as amniogenic cells. These are referred to as amniogenic cells. Clear? Now that epiblast is going to multiply and they are going to form a layer of cells here, and this layer of cells over here are going to be known as what, sir? Amniogenic cells. And because of this layer which is formed here, there is a formation of cavity here. And this cavity will be known as the amniotic cavity. Oh my God. It's so simple. आपने कई बार सुना होगा amniotic cavity. This is how amniotic cavity will be actually formed here, sir. So the epiblast cells are going to multiply to form your amniogenic cells, and because of that there is a cavity formed there. That cavity will be known as the amniotic cavity. Now similarly, what is going to happen? If epiblast is going to multiply, will the hypoblast keep quiet there? No, not at all. Not at all. So अब क्या होगा? Now what is going to happen, my dear friends? Even the hypoblast cells will also multiply right now. The hypoblast cells will also multiply, and they will be multiplying and forming a layer of cells over here. The hypoblast will also multiply, and that is going to form a layer of cells over here. Now this layer here, it is referred to as the Husser's membrane. It is referred to as what here, sir? Husser's membrane. और जैसे ही यहाँ पर ये Husser's membrane form होता है, the moment this Husser's membrane is going to form here, therefore what is going to happen, my dear friends? Yes, again this is also going to become like a cavity here, and this cavity will be known as the yolk sac. And of course, this one here will be the primary yolk sac. Primary yolk sac. It's a beautiful symmetrical thing, sir. In the second week, everything is like symmetrical here. तो धीरे धीरे आप देख रहे हो फर्दर डेवलपमेंट कैसे होते जा रहा है, हाउ द फर्दर डेवलपमेंट इज गोइंग टू टेक प्लेस. तो फर्स्ट विल बी हैविंग एपिब्लास्ट, देन दे विल बी हाइपोब्लास्ट, and then the epiblast will multiply to form amniogenic cells to form a cavity above amniotic cavity, and hypoblast will multiply to form Husser's membrane, and then again there is a cavity there. That cavity will be the primary yolk sac. Okay, now my dear friends, I'll tell you one very interesting thing here. We are in which week right now? We are in which week of the pregnancy right now, sir? We are in the second week of pregnancy. In the second week of pregnancy, you can clearly observe there, everything will be like double, sir. Everything will be like double. Second week, everything is double. Kaise, how? You can clearly see that. You can very clearly see here that there will be two layers, epiblast and hypoblast. Epiblast and hypoblast. There'll be two layers, and then again you'll be having like two cavities here, sir. One is your amniotic cavity, another one will be the yolk sac. Amniotic cavity and the yolk sac here, sir. Fine. And after that, there will be again two layers, that will be the amniogenic cells here and the Husser's membrane. So what I'm trying to tell you is that in the second week, there's a beautiful symmetry here. Everything will be like double, sir. Everything will be like double. So keep that in mind. That will help you in remembering very easily here. In second week, सब कुछ यहाँ पर ना double होता है. So there will be epiblast and hypoblast, two layers. Then there will be two cavities, above amniotic cavity, below yolk sac, and then there will be two layers. अब क्या होगा? Now what is going to happen? चलिए आगे बढ़ते हैं सर. After this, what is going to happen? Let us go to the third diagram right now. Now, my dear doctors, after this, what is going to happen is there will be formation of mesoderm. अब यहाँ पर mesoderm form होता है. I want you all to remember this or listen to this very carefully here. Now, why I'm telling you very carefully here, my dear friends, remember that कि अब क्या होता है, mesoderm form होता है. The moment I'm telling you that there is a formation of mesoderm here, the students are going to think that, okay, sir told that epiblast is going to become ectoderm and hypoblast is going to become endoderm, and then ectoderm endoderm अगर ये बन जाएगा तो इसके बीच में यहाँ पर mesoderm form होना चाहिए. Students will think by themselves that, अच्छा mesoderm should form here. But listen to me very carefully here. Whatever we have drawn here in the blue color and the red color here, my dear friend, that is your epiblast and hypoblast here. Whatever we have drawn in like epiblast and hypoblast here, that is your embryo proper. embryo proper. What is the meaning of that one? That is the actual embryo. The changes will keep on taking place in that one, and then it will develop into the future baby. So the actual future baby is going to be formed from this embryo proper, sir. Actual embryo proper here. Fine. So my dear friends, what is going to happen? If that is your embryo proper, whatever mesoderm is going to be formed inside that one, whatever mesoderm is going to form inside that one, that mesoderm will be known as the intra-embryonic mesoderm. Listen to me very carefully. That mesoderm will be known as intra-embryonic mesoderm. Is it okay? Then, apart from this intra-embryonic mesoderm which is going to form here, apart from that, there is one more mesoderm formed outside this embryo, embryo ke bahar. Outside the embryo, that is why it is known as what, guys? Extra-embryonic mesoderm. Extra embryonic mesoderm कहते हैं उसको, ठीक है ना? So one will be the intra-embryonic mesoderm, another one is the extra-embryonic mesoderm. So clear? So there will be like two types of mesoderm here. One is intra-embryonic mesoderm, another one is extra-embryonic. Okay. Please listen to me very carefully, sir. There is a reason why I'm repeating it like double times here, क्योंकि यहीं पर तो बहुत सारे स्टूडेंट कंफ्यूज होते हैं. अब यहाँ पर क्या होता है, जो इंट्रा एम्ब्रियोनिक मेसोडर्म होगा ना, इंट्रा एम्ब्रियोनिक इनसाइड द एम्ब्रियो, दैट विल बी फॉर्मिंग इन विच वीक? थर्ड वीक. That is going to form in which week? Third week. Why? Because I told you very clearly, in third week there will be the formation of 3 germ layers. In third week there will be formation of 3 germ layers, ectoderm, endoderm and mesoderm. तो वो तो third week में form होगा. But now we are in which week? We are in second week. और second week में कौन सा वाला mesoderm form होता है? That is extra embryonic mesoderm. Please remember, that is extra embryonic mesoderm. ठीक है? Let us repeat once more neat and clearly. Mesoderm will be of two types. There will be two types of mesoderm. One is inside the embryo, intra-embryonic mesoderm, and one is outside the embryo, that is extra-embryonic mesoderm. Intra-embryonic and extra-embryonic will be there. Intra-embryonic mesoderm will be formed in which week, sir? It will be formed in third week. Whereas extra-embryonic mesoderm will be formed in which week? That will be formed in the second week. Okay. That is the only thing you have to remember. अगर आपको एक फाइनल कंक्लूजन चाहिए, सर मुझे ज़्यादा कुछ कहानी नहीं चाहिए, आई जस्ट वांट टू रिमेंबर दैट वन. याद रखिए, इंट्रा एम्ब्रियोनिक विल बी फॉर्मिंग इन थर्ड वीक, वेयर एज एक्स्ट्रा एम्ब्रियोनिक mesoderm विल बी फॉर्मिंग इन द सेकंड वीक. तो वी आर इन विच वीक राइट नाउ? सेकंड वीक. तो चलिए आगे बढ़ते हैं सर, नेक्स्ट डायग्राम. अब क्या करेंगे हम? What we are going to do is, we'll not be able to draw this diagram again and again. There are like so many cells in this one. तो ये सारे cells हम ना दोबारा ड्रॉ नहीं कर पाएंगे. तो let me make the diagram simple for you. How? Please look here. Now, for example, imagine this blue colored line to be the layer above. Epiblast. Epiblast will multiply to form amniogenic cells, and this cavity here is amniotic cavity. Now, this red colored layer below here will be the hypoblast, and that hypoblast is going to multiply and form Husser's membrane. Husser's membrane, and this cavity below here will be the yolk sac. क्या बात है? Understanding? And I told you very clearly, the outer black colored line here will be the trophoblast. The outer black colored line here will be the trophoblast. Clear with this one here? So the same diagram here, but we are not going to draw that cells again and again. It will take a lot of time here, my dear friends. So therefore, now what we are going to do is just draw a simple line diagram here. अब मैंने आपको क्या कहा था? This blue colored and red colored one, epiblast and hypoblast, is embryo proper, actual embryo. But if that is actual embryo, the mesoderm inside that intra-embryonic mesoderm, outside that will be the extra-embryonic mesoderm. Okay. तो अब क्या होगा? Now what is going to happen? There will be formation of mesoderm here. There will be formation of mesoderm outside the embryo here, sir. So whatever I'm highlighting here in yellow color, even that is also like cells only. Whatever I'm highlighting in yellow color there, that is also like cells only. But those many cells we are not going to draw right now, my dear friends. Please remember that it is extra embryonic mesoderm. आप ऐसे मान लो कि ये वाला यहाँ पर extra embryonic mesoderm है. Is it okay? So this one will be the extra embryonic mesoderm, sir. EEM. Now in the exam, the question is all about what? It is derived from where? So remember, these cells are going to be coming from the yolk sac. The extra embryonic cells are actually, extra embryonic mesoderm will be derived from where? It is derived from primary yolk sac. Now, why I'm telling you this point here, my dear friends, please remember, in the old edition of embryology textbooks, it was given that it is derived from the trophoblast. Please remember that it is not derived from the trophoblast. According to the latest edition of Langman's embryology, I'm telling you, and from the Langman's embryology, I'm telling you that it is derived from where, sir? Primary yolk sac. So the extra embryonic mesoderm is actually derived from the primary yolk sac. Primary yolk sac. I hope the things are very much clear now. Come on, my dear doctors, is it clear? So I hope you're understanding my simple diagrams here, so line diagrams here. So therefore, now what is going to happen? Extra embryonic mesoderm, and that will be derived from primary yolk sac. Ab kya hoga? Now what is going to happen? The last and final, I told you I'll be telling you in 4 diagrams here, sir. Now welcome to the 4th diagram here. Ab kya hoga? Now we are in which week right now, sir? 2nd week. And 2nd week, my dear doctors, remember, I told you everything has to be like double. सेकेंड वीक में तो सब कुछ डबल डबल होना चाहिए. तो हाउ कम दिस एक्स्ट्रा एम्ब्रियोनिक मेसोडर्म कैन बी अ सिंगल लेयर, सर? हाउ कैन दिस एक्स्ट्रा एम्ब्रियोनिक मेसोडर्म कैन बी अ सिंगल लेयर? वो भी तो डिवाइड होना चाहिए. या इट मेक सेंस, है ना? सो देयरफॉर माय डियर फ्रेंड्स, रिमेंबर, in this extra embryonic mesoderm there will be formation of coelom. There will be formation of a coelom, sir. A cavity will be formed here. And of course, that cavity will be known as what, guys? Extra embryonic coelom. And because of that coelom, it is going to divide the entire extra embryonic mesoderm into 2 layers. Because of that coelom inside, it is going to divide into how many layers there, sir? Like 2 layers. Fine. So now, welcome here. Tell me for the last and final time right now, what is this blue colored layer above? Epiblast. That is going to multiply to form amniogenic cells, and this cavity will be the amniotic cavity. Now, what is this red colored layer cells below here? Hypoblast, and that will be multiplying to form Husser's membrane, and this cavity here will be yolk sac. And this black colored line on the outer side here will be the trophoblast. मैंने आपको कहा था ना, till the last I'll be using the same colors. It will be easy for you. Now, outside whatever space you have, that is extra embryonic mesoderm, and in this mesoderm, what is going to happen here, sir? There is a formation of a cavity. And this coelom is known as the extra embryonic coelom. And because of this coelom, what is going to happen? The entire mesoderm here will be divided into 2 layers. Wow. Are you understanding the clarity right now? I hope now you're getting the clarity, my dear friends. So this coelom here will be the extra embryonic coelom. This coelom here will be the extra embryonic coelom. Okay. That is extra embryonic coelom. And because of that extra embryonic coelom, what is going to happen? The mesoderm will be divided into 2 parts here. 2 layers here, sir. One on the inner side, one on the outer side here, sir. So because of that extra embryonic coelom, what is going to happen? The mesoderm will be divided into 2 layers on the inner side and outer side. Now, my dear doctors, yes, if you are telling that it is like inner outer, it is absolutely wrong. अगर आप अंदर बाहर करके बोलोगे ना, like there are 2 layers on the inner side and outer side, absolutely wrong. It is wrong. Why? Why? Because you can clearly see that this mesoderm, which I'm telling like inner one, it is actually in continuation here. And this mesoderm, which I'm telling the outer one, it is also in continuation here. So we want the exact landmark. We want the exact landmark. Okay, where exactly is the like 2 mesoderms are present, sir. So please remember, first of all, the name here. The inner one here will be known as the splanchno. The inner one here will be known as the splanchno. Pleuric. Extra embryonic mesoderm. And the outer one here will be known as the somato. Pleuric. Extra embryonic mesoderm. So inner one will be the splanchno-pleuric extraembryonic mesoderm. The outer one will be the somato-pleuric extraembryonic mesoderm. So we'll be having splanchno-pleuric and somato-pleuric. Fine. And these words are going to come like again and again, my dear friends. Don't worry about this word, that is somato and splanchno. In embryology, wherever you are going to come across, like, in embryology, wherever you are going to come across somato is representing what here, sir? The body wall. So it is present always along with the outer part. body wall apart, and splanchno is representing the visceral organs there, so it will be towards the viscera. The somatopleuric will be along with the body wall, whereas splanchnopluric will be along with the viscera. That is very clear. But now again, my question still remains there. Sir, as a simple medical student in first year, I'm getting confused here. You're telling this is the inner one and this is the outer one here, but I'm getting confused here. It is in continuation, sir. This is in continuation here. So where is the exact landmark? Welcome to Langman's embryology. It has given very, very clearly that only this mesoderm which is around the yolk sac, अराउंड द yolk sac, मतलब यहां से लेकर यहां तक, only this mesoderm which is around the yolk sac here will be nothing but the splanchnopluric extraembryonic mesoderm. Remaining everything will be somatopleuric, sir. तो प्लीज राइट डाउन देयर वेरी क्लियरली, स्प्लैंक्नोप्ल्यूरिक मतलब कौन सा वाला मेसोडर्म होगा, विच इज ओनली एंड ओनली अराउंड द yolk sac. That's all. The mesoderm which is present only around the yolk sac will be the splanchnopluric. Remaining everything will be the somatopleuric. Are you getting the clarity now? Hello my dear doctors, are you getting the clarity right now? अगर यहाँ से आपको clarity नहीं मिल रहा है तो I can do one thing for you. I can just highlight this with another color here. Now this mesoderm here which I'm highlighting here in green color, yes, that is the one which will be splanchnopluric extraembryonic mesoderms. Crystal clear concept. Okay, the splanchnopluric will be only that mesoderm which is around the yolk sac. Remaining everything will be the somatopleuric extraembryonic mesoderm. Okay, are you clear with this one here, sir? Now you might be thinking that why sir is like stressing a lot on this point. I told you like 3-4 times till now. Why I'm stressing a lot on this? Welcome here. Have you all heard about amnion and chorion, my dear friends? आपने कभी ना कभी तो सुना होगा ना amnion and chorion. तो please remember these two layers together, they are going to form your amnion. अब आप ना एक काम करो सर, just look at my diagram and tell me amnion will be formed by, just look at my diagram and tell me amnion will be formed by, diagram देखकर बताओ. तो what is that blue colored line? That blue colored line will be the amniogenic cells. Wow. What is that yellow colored one? The yellow colored one will be the somatopleuric extraembryonic mesoderm. Are you understanding now, Dr. Hamid? The amniogenic cells along with the somatopleuric extraembryonic mesoderm will be forming your amnion. That is forming amnion. इसी के लिए तो मैं आपको बार बार रिपीट कर रहा था कि you should have a clarity. It is not the splanchno वाला, ये splanchnopluric नहीं होता है, वो somatopleuric होता है. Okay, the next one, what is chorion? If that is your amnion, these two layers together they are going to form your chorion here. No need to mug up, sir. Welcome to my class. Look at my diagram and try to answer here. Chorion will be formed by dash and dash. Come on, tell me. So remember the outer black colored line here will be the trophoblast. And then the yellow colored one here will be the somatopleuric extraembryonic mesoderm. Are you understanding me? Chorion along with the somatopleuric extraembryonic mesoderm. Chorion is nothing but trophoblast along with the somatopleuric extraembryonic mesoderm. Perfectly done. So by this we are done with 2 more MCQs here, or 2 more things here. That is, one is your amnion here, another one will be the chorion here, sir. Even these 2 things also done here. Wow. Are you understanding me? And if you are done till here, my dear friends, remember by this we are actually done with what here? We are done with our second week of development, sir. Okay, second week यहाँ पर complete हो गया है. तो जैसे मैंने आपको कहा था, I am going to give you like 4 diagrams here. The first one here, the second one here, the third one here, and the fourth diagram here, sir. In these 4 diagrams, we are done with the second week. There's so many changes taking place in the second week of development. Are you clear with this one here? Now, shall we go towards the third week right now, sir? Hello, my dear doctors. Are you all alive? Just we are done with till the second week only right now. Okay. तो हम एक काम करते हैं सर, before going to the third week almost will be done in 5-10 minutes and we can wind up. But before going to the third week, let us do one thing. एक बार हम लोग ना इसको रिवाइज कर लेते हैं from the day 1. एक बार रिवाइज करते हैं सर. Let us revise from day one. Okay, now let's see like what is are we able to like remember that in the sequence there? Fine. So I want you all to revise along with me right now, my dear friends. Number one, what is happening on day one? Day one पर क्या होता है? Fertilization होता है. Now, what is the result of fertilization? The result of fertilization is zygote. Zygote will multiply to form what? Morula. How many cells? 16 cells. That will multiply to form late morula. How many cells? 32 cells. And that will enter into uterus. Now, after entering into the uterus, that is going to become what? Blastocyst. And that blastocyst will undergo implantation. अब जैसे ही वो ब्लास्टोसिस्ट इम्प्लांटेशन होगा, लाइक व्हाट इज द स्ट्रक्चर? ब्लास्टोसिस्ट में क्या होता है? आउटर सेल मास होता है, इनर सेल मास होता है, एंड देयर इज अ फ्लूइड फिल्ड कैविटी. There is outer cell mass, inner cell mass, and fluid filled cavity. Okay. The moment that blastocyst is getting implanted, the outer cell mass is going to become, the moment the blastocyst is implanted, the outer cell mass is going to become what, sir? Trophoblast. The outer cell mass is going to become trophoblast. Then, sir, what about the inner cell mass? Inner cell mass, only one layer is modified into hypoblast, and the remaining cells are going to become what, sir? Epiblast. समझ में आ रहा है? If you look at my diagrams over there, they're like an animation, sir. तो inner cell mass is going to form epiblast and the hypoblast. अब आगे क्या होगा? Epiblast will multiply to form amniogenic cells, cavity, amniotic cavity. Hypoblast will actually multiply to form Husser's membrane. There is a cavity, that cavity is yolk sac. अब आगे क्या होता है? What is going to happen further? Embryo के बाहर, outside the embryo, there is a formation of mesoderm. Extra embryonic mesoderm form होता है. And that is derived from where? Again, yolk sac only. Primary yolk sac cells will multiply and form that extraembryonic mesoderm. And now inside that, there is a formation of coelom, and that coelom will be known as extraembryonic coelom here. And because of that, it is going to divide again into two layers. The mesoderm, which is surrounding only the yolk sac, the mesoderm only around the yolk sac here, that is splanchnopluric extraembryonic mesoderm. And the remaining mesoderm on the outer side, there will be somatopleuric extraembryonic mesoderm. That is somatopleuric extraembryonic mesoderm. Are you clear with this one here? सर आपसे पढ़कर बहुत ईजी लग रहा है टॉपिक. थैंक यू थैंक यू थैंक यू. मैंने आपको प्रॉमिस किया था ना, लाइक एम्ब्रियोलॉजी विल मेक इट डैम ईजी. तो यू जस्ट ड्रॉ द डायग्राम्स अलॉन्ग विद मी. ऑटोमेटिकली यू कैन राइट दैट आंसर इन इंग्लिश देयर. आप आराम से उसको इंग्लिश में लिख सकते हो. इट्स ऑल अबाउट अंडरस्टैंडिंग द कॉन्सेप्ट हियर. क्लियर? नाउ वेलकम टू थर्ड वीक सर. अब आ जाइए थर्ड वीक में, ठीक है ना. इन थर्ड वीक द गुड न्यूज हियर इज दैट there will be one change taking place in epiblast. There will be a one change taking place in epiblast here, and there will be one change taking place in hypoblast. मतलब थर्ड वीक में क्या होता है? In third week, basically the changes are taking place only and only in the epiblast and hypoblast. Only these two only. Only epiblast and hypoblast only. Okay. One change in epiblast, one change in hypoblast. Therefore, what we are going to do, we are not going to draw this entire diagram again and again, sir. ये पूरा diagram हम दोबारा draw नहीं करेंगे. We are not going to draw this diagram entirely. Okay. So what we'll do from this diagram, from this entire big diagram here, we are just going to remove that epiblast and hypoblast from here, sir. So this epiblast and the hypoblast, these two layers, we are going to remove from here, and we'll see what is the change taking place in epiblast and what is the change taking place in hypoblast. The one change in epiblast and one change in hypoblast, we'll see that changes separately. Clear with this one here? Are you getting a point here? Now we are not, don't think that everything else is gone, sir. All the remaining parts are present there. Fine. All these remaining parts are already present there. वो सब कुछ होता है वहाँ पर, ठीक है ना. वो नहीं जाएगा. तो हम क्या करेंगे, बस यहाँ से एपिब्लास्ट और हाइपोब्लास्ट बाहर निकालते हैं और उसको स्टडी करते हैं, ठीक है. तो लेट्स रिमूव दैट वन एंड स्टडी ओनली व्हाट आर द चेंजेस टेकिंग प्लेस इन थर्ड वीक. वेलकम टू थर्ड वीक नाउ. Now in third week, what is going to happen? As usual, our habit, हमेशा क्या पढ़ना चाहिए सर? Always you have to know before starting the week what is happening there. In third week, what is going to happen? Gastrulation will take place. Gastrulation will take place, sir. Now in gastrulation, what is the meaning of this one? It means formation of 3 germ layers. Gastrulation का मतलब क्या होता है सर? Formation of the 3 germ layers. Ectoderm, endoderm and the mesoderm. Formation of the 3 germ layers is known as what? Gastrulation. Now I'm going to use the same colors till the end. The blue colored line above will be hypoblast. Sorry, the blue colored line above will be the epiblast, and this red colored line here will be the hypoblast. Are you understanding? तो मैंने वहां से एपिब्लास्ट और हाइपोब्लास्ट निकाल दिया सर. So from here, epiblast and hypoblast, we have removed that out. So epiblast and hypoblast. Now one change in epiblast, one change in hypoblast. That's all. Now tell me, in hypoblast, there will be normally cube-shaped cells. Now this cube-shaped cells here, they are going to modify into tall and columnar. And the remaining cells will be remaining like cube-shaped only. Only few cells will modify into tall and columnar cells. Remaining will be like as normal as cuboidal cells only. Now, once these cells are going to modify into tall and columnar cells, इसी को हम कहते हैं prochordal plate. This itself is referred to as what, guys? Prochordal plate. Is it clear? So in hypoblast, some of the cells are modified into tall and columnar, so that is known as prochordal plate. Now, sir, we understood the name. Okay, it is known as the prochordal plate. Fine. But what is the function of this one? What is the use of this one? Okay. Now, my dear friends, remember, procaudal plate is an indication that, acha, on this side, the future head of the baby is going to form. तो wherever procaudal plate is forming in the embryo, in that embryo, जहां कहीं पर भी procaudal plate form होता है, उस side में future head develop होगा sir. Head of the baby is going to form. Okay, if head is going to form here, this is going to become which end? Cranial end. And obviously the opposite end is going to become which end here? This is going to be caudal end. Clear? So therefore, procaudal plate is the one which is going to determine, it is going to decide craniocaudal axis of embryo. Oh ho. तो बाय द एंड ऑफ सेकेंड वीक ऑल्सो हमको पता नहीं चलता है सर. ओके ना, अगर आप ये डायग्राम देखोगे, दैट इज लाइक एंड ऑफ सेकेंड वीक, एंड टिल द एंड ऑफ सेकेंड वीक ऑल्सो वी डोंट नो वेयर इज द हेड ऑफ द बेबी, वेयर इज द टेल ऑफ द बेबी. यू डोंट नो द क्रेनियल एंड कॉडल एंड, नहीं पता चलता है. नाउ वेलकम टू द थर्ड वीक. इन थर्ड वीक देयर इज अ फॉर्मेशन ऑफ प्रोकॉडल प्लेट, एंड दैट प्रोकॉडल प्लेट इंडिकेट्स दैट हियर द फ्यूचर हेड ऑफ द बेबी इज गोइंग टू फॉर्म. ओके, तो मान लो कि लाइक दिस वन इज योर हाइपोब्लास्ट हियर, और इस हाइपोब्लास्ट में मान लो कि यहाँ पर ये ना सेल्स ऐसे ना, like out of all the cube shaped cells, some of the cells are going to become like sir columnar. तो जैसे यहाँ पर procaudal plate form होगा, okay, that means on this side what is going to happen? The head of the future head of the baby is going to form. So this end is going to become the cranial end. Cranial end. Understanding? So therefore, my dear friends, this concept is very much important. So which structure is going to decide the craniocaudal axis of the embryo? Craniocaudal axis of the embryo, that is procaudal plate. Procaudal plate. Is it okay? Now after learning about the hypoblast, now let us see about the epiblast right now, sir. Epiblast में क्या change होता है? What is the change in epiblast? My dear friends, are you understanding? I hope I'm not confusing you. More than you people, actually, I'm fearing here that I should not confuse you actually, because embryology ना एक वो norm बना हुआ है among the students. The seniors will be conveying to the juniors that अरे embryology बहुत difficult है, difficult है. ऐसा कुछ नहीं है sir. It's all about understanding properly. Okay. So if you understand आराम से once properly and everything will be clear. I hope I'm not confusing you more. I hope the concepts are getting cleared. तो procaudal plate clear हो चुका है. Who is going to decide the craniocaudal axis of the embryo? That is procaudal plate. So done. And now welcome to epiblast right now, sir. In epiblast, अब थोड़ा सा ध्यान से सुनना sir. This is the last thing. Hello. This is the last thing, but this is going to be little bit confusing. थोड़ा सा ध्यान से सुनना. Full concentration here. Don't look here and there, my dear friends. Don't concentrate on other things, sir. Another like 5-10 minutes. Completely be with me, sir. बस आप, मैं और अपनी anatomy. ठीक है ना? Epiblast, some of the cells of epiblast from the caudal end will multiply and move forward. Caudal end से यहाँ से multiply होगा ना, आगे बढ़ते हैं. ठीक है ना? So the cells are multiplying and moving forward, 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 forward. And suddenly in the middle there is a formation of big mass of cell. Okay, and the remaining cells are going to remain normal like this only, like tall and columnar cells. Fine. So epiblast में क्या होता है? Some of the cells will multiply and move forward. From the caudal end, they're moving towards the cranial end, moving, and in the middle they're going to form a dense mass of cells. Okay. पहले आप आराम से इसका नाम सीख लो. What is the name? Please remember, these cells which are multiplying, proliferating and moving forward, these cells are referred to as primitive streak. This is primitive streaks. ठीक है ना? And this dense mass of cells in the middle, which is formed here, this one will be known as the primitive knot. KNOT knot. And this primitive knot, it is also known as Hensen's node. So the cells which are multiplying and moving forward will be the primitive streak cells, and the cells in the middle which are going to form a dense mass of the cells, that is known as the primitive knot. Primitive knot form होता है. ठीक है ना? Now once you have learned the names, now my dear friends, like what is the function of this one? Now let me tell you first of all what is the function of primitive streak. Primitive streak का function क्या होगा? ठीक है ना? Now, are you listening to me carefully, sir? Please listen this very carefully. I'm telling you again and again. Okay, na? Primitive streak का काम क्या होता है? In primitive streak, what is going to happen? Certain cells will multiply to form a group of cells. And this group of cells, what is going to happen? They will move down. After multiplying, that group of cells are going to move down. What is the layer below? Below, you're able to see that red colored one, hypoblast. And after that, cells are going to move down. They will replace all the old cells of hypoblast with new cells. It is like renovation. It is like renovation. All the old cells of hypoblast will be removed and they will be taken care by the new cells from here. Okay. And now try to recall. मैंने आपको क्या बताया था? What is another name of hypoblast? Hypoblast is also referred to as the primitive endoderm. Primitive endoderm. And now once it is replaced by new cells from here, it's no more primitive, it's now definitive endoderm. The new endoderm layer is formed here. Beautiful story. And after that, another group of cells will multiply and they will move in the middle here, and that group of cells are going to form mesoderm. And after that, another group of cells will multiply and they are going to replace their own layer. Which layer? Epiblast layer. And all the old cells will be removed and they'll be replaced by the new cells over there. And therefore, my dear friends, yes, that is going to become your ectoderm. It is going to form what's a ectoderm. Fine. So what is going to happen here? The primitive streak cells, I'm going to repeat this again here, my dear friends. The primitive streak cells, what is going to happen? One group of cells are going to replace all these old cells in hypoblast to form the definitive endoderm. And then these cells are going to multiply and form the mesoderm. And they themselves are going to multiply and form the ectoderm. Okay. It means today we are actually learning that, all the three germ layers are formed from where? All the three germ layers are derived from where? Okay, primitive streak. Primitive streak forms all three germ layers. Oh my God. Right from school days and 11th and 12th standard, we have been learning that three germ layer, ectoderm, endoderm, and mesoderm. Today we have learned that all the three germ layers are derived from where? They are derived from primitive streak. Are you understanding me? Not only that, we also learned today the sequence of formation of germ layers. मैंने आपको क्या बताया था? सबसे पहले कौन सा वाला फॉर्म होगा? एंडोडर्म. That will be followed by mesoderm, and that will be actually followed by the ectoderms. पहले एंडोडर्म फॉर्म होता है, फिर mesoderm फॉर्म होता है, फिर ectoderm फॉर्म होता है. Crystal clear concept. So from today onwards, remember what should be your answer? All three germ layers are derived from primitive streak, and the sequence of formation first endo, then meso, and then ectoderm. Perfectly done. आगे बढ़ूं मैं? Should I go ahead? And now, what is the function of the primitive knot? Primitive knot का function क्या होगा? Now in primitive knot, again the same thing. Some of the cells will multiply and they move down, and they're going to move forward. Is it okay? And they will be stopping just before the procaudal plate. Procaudal plate से वो पहले रुक जाते हैं. And this cells which I am drawing in black color here, these cells which I am drawing in black color here, don't think that it is like a layer of cells. No, it is not like a layer. It is just like a cord, like my pen here. Okay, it's like a cord of the cells, and therefore it is known as notochord. इसी को हम कहते हैं notochord. So remember, if anyone is going to ask you, can notochord will be derived from where? Remember, it is derived from primitive knot. So what is the function of primitive knot? It forms notochord. Primitive knot is going to form the notochord, sir. Lovely. I hope it is clear. By this, actually, our third week is clear here, sir. And remember, like this, it is like very much symmetrical. In second week, everything is like double. In third week, as a student, I'm going to remember like three things, sir. In third week, number one, we have to remember. Come on. In third week, number one, we have to remember about procaudal plate. It is going to decide the craniocaudal axis. Number two, we have to remember about the primitive streak. It is going to form all three germ layers. And number three, we have to remember the primitive knot. It forms the notochord, sir. That's all. That's all. सो सेकेंड वीक में सब कुछ डबल डबल और थर्ड वीक में ट्रिपल. तीन चीजें याद रख लो, कहानी खत्म. Remember these three things, that is done, sir. So which structure is going to decide the craniocaudal axis of the embryo? That is nothing but procaudal plate. Which structure is going to form all three germ layers? That is primitive streak. And which structure is going to form the notochord? Notochord is formed by primitive knots. That's all. Done. Come on, my dear doctors. Are you clear with this one here? Are you all clear with this one here? Should I ask you one question here? Thank you. Thank you. It's clear now. Yeah. Should I ask you one question here, my dear friends? Can you please look at this diagram here of the embryo and tell me, okay, this diagram of the embryo is in longitudinal section or transverse section? Is it longitudinal section or transverse sections? This diagram of the embryo on the screen, देख कर बताओ सर, आप भी क्या याद रखोगे? Is it longitudinal section or transverse section? लॉन्गिट्यूड ये बढ़िया है आप लोग का ना. ये सही बात है. मैं आपको दो ऑप्शन देता हूँ, आप मुझे वापस दो ऑप्शन देते हो, है ना. मैं पूछ रहा हूँ लॉन्गिट्यूडनल ट्रांसवर्स, सम पीपल आर टेलिंग ट्रांसवर्स, सम पीपल आर टेलिंग लॉन्गिट्यूडनल. सही बात है भाई. और थोड़े लोग को मैं अपने से देख सकता हूँ कि आफ्टर टेलिंग द आंसर वो लोग ना टशन में बैठे हैं. अच्छा चलो अब देखते हैं सर क्या बताते हैं. तो दो ऑप्शन मैं आपको देता हूँ, दो ऑप्शन आप मुझे वापस देते हो. बताइए लॉन्गिट्यूडनल या ट्रांसवर्स है. लिसन हियर, आई विल टेल यू. फाइन. सब लोग मुझे यहाँ पर ध्यान से देखो. आई विल नॉट बी एबल टू रिपीट इट अगेन एंड अगेन. लुक एट मी फॉर वंस एंड फॉर एवर. फाइन. Now, for example, if my this hand here, if it is visible, दिखाई दे रहा है? हाँ. My this hand here is your endoderm, so that red colored one. This is endoderm, red colored one. And now my this hand here will be the ectoderm here, sir. And in the middle you're having the mesoderm. And mesoderm, you imagine there's a cord-like structure passing from here. That is your notochord. My pen here is like cord. It's like notochord here, sir. Notochord is passing in the middle. Now endoderm here, ectoderm here, sir. And in the middle you're having mesoderm. And then there is a notochord passing from there. This is the embryo. So if you understand the embryo, remember in three dimension, 3D here, this is the embryo here. This is the embryo. Now this embryo, I am cutting it longitudinally. And after cutting it longitudinally, we are seeing from a side here, sir. हम लोग ना ऐसे side से देख रहे हैं. So when you're looking from a side here, okay, ectoderm, endoderm, mesoderm, and the notochord is visible. So exactly that is the diagram. That is the longitudinal section, sir. Understanding here? It is the longitudinal section here. So this diagram is actually representing the longitudinal section. अब हम क्या करेंगे इसको transversely ऐसे cut करेंगे. So we are going to cut it like this transversely. And after cutting transversely, I'm turning towards you like this. And once I turn it toward you, what is this hand here? Endoderm. What is this one here? Ectoderm, mesoderm, and this is the notochord. And in this diagram, you'll not be able to see the entire notochord. Only this much part is visible. Only this much part. In the longitudinal section, side से हम देख रहे हैं तो पूरा notochord नजर आएगा. You'll be able to see the entire notochord. But here, only this much part of the notochord is visible there. Understanding? There is a transverse section. So therefore, this diagram on the screen here, this is actually the longitudinal section. Longitudinal section, my dear friends. Yes, very good Anish, very good Hamid, Shirin. Very nice, very nice. Okay. तो अब हम draw करेंगे transverse section of the embryo. So please write on the heading there as transverse section of embryos. Transverse section of the embryo. Now in the transverse section, again the same thing. This one here will be the ectoderm here, and this one here will be the endoderm here, and this here will be the notochord. Okay. Understanding now? So this is your ectoderm, and this one will be the endoderm, and this one is mesoderm in the middle. And yes, passing through that one will be the notochord. अगर आप आज से ऐसा डायग्राम देखोगे तो please remember my dear friend, yes, it is going to be the transverse section of embryo. Okay. Now let me ask you one more question here, sir. We are almost at the end of the class now. That is why I'm asking you the question to understand whether you have understood my class or not. Okay. So can you please tell me which Which mesoderm is this one here, sir? ये कौन सा वाला mesoderm है? Is it like intra-embryonic mesoderm or extra-embryonic mesoderm? इंट्रा-एम्ब्रियोनिक, एक्स्ट्रा-एम्ब्रियोनिक, कहानी याद है? मैंने आपको कहानी बताई थी पूरी डिटेल में. Is it intra-embryonic or extra-embryonic mesoderm? Yes, very good, Dr. Anish. It is intra-embryonic. मैंने आपको क्या कहा था? Please remember, extra-embryonic mesoderm will be formed in second week. Intra-embryonic will be formed in which week? Third week. और ये embryo के अंदर है, तो ये intra-embryonic mesoderm है. Okay. And this intra-embryonic mesoderm is in turn divided into three parts. So this one here will be the first part. Okay. This is the first part here, and then you have got the second part here, and then you'll be having the third part here. Intra-embryonic mesoderm is in turn divided into like three parts here, the first part as well as the second part here and third part here. Is it okay? Now, what is the name of this one here? And my dear friends, do you remember our logic? Hello? मैंने आपको क्या कहा था? Second week everything is double double, third week everything is triple double. Even now it is clear. Now also it is true here कि mesoderm कितने parts में divide होते हैं यहाँ पर? 3 parts में divide. Everything triple triple. Now this one is along the axis. So therefore this one is para-axial mesoderm. Para-axial mesoderm, sir. Para means along the axis. Para-axial mesoderm. And this second one here, it is actually in the middle. So therefore it is known as the intermediate mesoderm. Very meaningful name. And then finally, my dear friend, the third one here will be on the lateral side. So therefore this is known as the lateral plate mesoderm. Are you understanding me? So in the third week, what is going to happen? The intra-embryonic mesoderm is divided into three parts. Along the central axis, that will be para-axial mesoderm. Then in the middle, it is intermediate mesoderm. And on the lateral side, it is lateral plate mesoderm. Is it okay? और अब फर्दर मतलब हमको बस इसके ना डेरिवेटिव्स पढ़ने होंगे. यू हैव टू एक्चुअली लर्न कि सोमाइट्स कहाँ से फॉर्म होते हैं, स्केलेटल मसल, स्मूथ मसल, कार्डियक मसल, यूरोजेनिटल सिस्टम एंड ऑल दोस थिंग्स, उसके डेरिवेटिव्स होते हैं सर. फाइन, वी नीड टू जस्ट लर्न द डेरिवेटिव्स ऑफ दैट वन. क्लियर? नाउ लेट मी शो यू वन पिक्चर हियर. Hello, my dear friends. Let me show you one picture here, sir. Please look at this diagram here. Of course, my dear doctors, like you know very well, general embryology, it can't be just completed in a span of 1.5 hour. तो इसीलिए like I'm not going in detail of now what are the derivatives from here. I hope you got the entire scenario very clear now. What is happening in first week, second week and third week. वो तो बड़ा clear हो गया है ना? That is very much clear now. So later on we'll be studying about their derivatives and all those things. That is a separate story. Is it okay? So I'm not going in detail of the somites, then dermatome, sclerotome, myotome, बहुत कुछ होता है ऐसे derivatives. First you understand the main thing. Okay. If you have understood the main concept here, that will be done. Otherwise, normally general embryology will take approximately like 2.5 hours to 3 hours of time. तो 3 घंटे में जनरल एम्ब्रियोलॉजी पूरा कंप्लीट हो जाता है. एवरीथिंग विल बी डन. फाइन. तो अभी के लिए आई थिंक दिस इज मच इनफ. नाउ प्लीज लुक एट दिस डायग्राम हियर. The moment you look at this diagram here, you can clearly identify, yes, it is transverse section of embryo. Now the only and only thing I want you is I'll mark here and you will identify this one, sir. Okay ना? मैं mark करता जाऊंगा, आप identify करते जाइए, sir. Now if this one here will be the transverse section of the embryo, what is this germ layer on the outer side here? Please keep on answering me. What is this germ layer on the outer side? This one here will be the ectoderm here. Now what is this germ layer on the inner side here? This one here will be the endoderm. And in the middle, you'll be having the mesoderm. Now, is it a longitudinal section or transverse section? Yes, of course, it is actually the transverse section. Sir, how can you say it is transverse section? You can very clearly see the notochord here, sir. Notochord. In transverse section, there is a notochord visible. Now, if that is a notochord here, what is that tube-like structure above the notochord, my dear friend? What is a tube-like structure above the notochord? What is this structure? So remember, that is actually the neural tube. Oh my God, ये neural tube है. What is that one? There is a neural tube, and that neural tube is the one which is going to form your central nervous system. That is your brain, and the spinal cord will be derived from that neural tube. तो नोटोकॉर्ड के ऊपर जो नजर आ रहा है आपको न्यूरल ट्यूब होता है, एंड न्यूरल ट्यूब इज द वन विच इज गोइंग टू फॉर्म योर ब्रेन स्पाइनल कॉर्ड. तो मैंने आपको क्लास के स्टार्टिंग में क्या बताया था, व्हाट आई टेल यू इन द वेरी बिगिनिंग ऑफ द क्लास, I told you that after third week, what is going to happen? Systemic embryology will begin. So in systemic embryology, I'll teach you how come that notochord above that one neural tube is going to form. उसके ऊपर न्यूरल ट्यूब कैसे फॉर्म होता है वो हम सेंट्रल नर्वस सिस्टम एम्ब्रियोलॉजी में पढ़ेंगे, सिस्टम्स में पढ़ेंगे, ठीक है ना. परफेक्टली फाइन, अवर प्लानिंग. नाउ व्हाट अबाउट द मेसोडर्म हियर. अब ये जो मेसोडर्म है, दिस मेसोडर्म इज द इंट्राएम्ब्रियोनिक मेसोडर्म. And that intra-embryonic mesoderm is going to divide into three parts here. You can see it very, very clearly here, my dear friends. Number one, this one here will be the para-axial mesoderm. Number two, this small one here, this one here will be the intermediate mesoderm. And finally, this one here will be the lateral plate mesoderm. I hope you're able to understand this very clearly now. Yes, my dear doctors, I think you're able to see it very clearly right now. The mesoderm is divided into three parts there: paraxial mesoderm, intermediate mesoderm, and the lateral plate mesoderm. That is intraembryonic mesoderm there, guys. Okay. So from today onwards, the moment you look at this diagram here, remember that is the transverse section of the embryo. Transverse section of the embryos. I hope now you're able to identify the diagram there. Clear? That is the transverse section of embryos. So I have given you our simple diagram also, as well as I have given you actual diagram also. And by this, my dear friends, as according to our planning, we are done with the first week, second week, and third week of the development. And by this, my dear friends, we are actually done with our general embryology. Of course, the derivatives of that one and all those things, we can do it separately again. And from here, we can actually begin with the systemic embryology, all the systems one by one. Sir. So did you all understand this one? So it's like a nice story, what is happening in first week, second week, and third week. Okay. So in this manner, you have to keep on learning with this one. I hope it is very clear. So just confirm me if this thing is very much clear, my dear friends. I hope the video will also be available. You'll be able to watch it later on also. So watch it again like once or twice and you'll be good to go. Okay. So thank you all. It was nice interacting with you all people. I hope I was able to remove that fear of embryology from you people. The embryology is not that difficult, my dear friends. It's like easy. You just have to follow all the diagrams sequentially one by one. Is it okay? Sure. Thank you all.